Good morning, biology class. Just trying to get this to focus a bit. That's better. Okay. All right, good morning, and this is a little discussion on the structure of DNA. DNA is a biomolecule like the proteins, like the carbs, and like the lipids. It's made of units, and the units are called nucleotides, and the nucleotides are arranged in a specific sequence. This note will also cover DNA's cousin RNA, which is a very similar structure. All right, so normally I do this in class where we cut out the nucleotide bases. And by the way, the nucleotide is made of 3 pieces, shown at the bottom. And we'll get to that in a minute. But the base of the nucleotide, it's called a nitrogen base, and it's this structure here, in this case. There are 4 different bases in DNA, and we're going to discover how they interact together first. So what I've done is I've got cutouts here, and I'll put them out. And what I've done is I've used color coding. Blue is delta minus and red is delta plus in a polar covalent bond. And there's a bunch of polar covalent bonds. I highlighted where the sugar attaches, and the sugar is a ribose sugar. In DNA, it's a deoxyribose sugar. All right, so what we're trying to discover is that which combinations will attract like little magnets. So let's take the 2 that are here, and look at that. I think they seem to work together. Red, blue, red, blue. Let's try the other small one to see. No, you see? Red, red, blue, blue, no good. Let's try the other big one. Red, red. There's one red, blue, but no, it's not going to work. So this is a pair. They'll stick together like a little magnet. Delta pluses and delta minuses will hold together the 2 nucleotides. And the force attracting them, of course, is a hydrogen bond. We learned about that in Unit 1. Let's see the other ones and see if they make a pair. I'll bring in the big one. And the small guy. And lo and behold, blue, red, blue, red, blue, red. 3 points of attachment. So these 2 are a pair. Each pair is made up of one larger one, a double ring structure, and one smaller one, a single ring structure. Larger ones are called purines, smaller ones are called pyrimidines, and we'll get to the specifics later on. Now, just to the general structure. Okay, so this is a typical nucleotide. Each nucleotide is made of 3 pieces. The nitrogen base, which I've outlined in blue, a 5-carbon sugar, which is here. 1, 2, 3, 4, 5 carbons. Oxygen is part of the ring. And that is the sugar. It's either ribose or deoxyribose. The nitrogen base. And the third part is called a phosphate. It's one of our functional groups. And it's over here. So a phosphate, a ribose sugar, and a nitrogen base. The most important thing to know about this nucleotide is the numbering system for the carbons. Just like in the other carbohydrates, this sugar has carbon numbers. And the numbers go like this. 1, tick. It's called 1 prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. There are 5 carbons in the sugar. 1 is always attached to the nitrogenous base. 5 always attached to the phosphate. In this particular sugar, on the number 2 carbon, we have a hydrogen. But there should be an alcohol group like number 3. This lack of an alcohol makes this a deoxyribose sugar, so this is a DNA nucleotide. DNA. RNA nucleotides instead have the alcohol group. The 2' carbon has OH, alcohol group. Let's take a closer look. So every nucleotide looks like this. Nitrogen base, 1'. This is a 2'. If it was a ribose RNA, it would be an OH. If it's a DNA, it's C with just an H. So that's one of the differences between RNA and DNA. So this is the 2' carbon, this is the 3' carbon, 4', 5'. Of all the carbons in the molecule, they all are important, but the ones that are most famous are the 3' and 5'. When you connect nucleotides together, the 3' of one nucleotide attaches to the phosphate on the 5' of the other. And that's how we make a polymer. Okay, DNA versus RNA table. First of all, the DNA structure is the famous double helix. It's 2 long strands intertwined in what they often call a twisted staircase. RNA is always single strand. DNA, as I said before, has a deoxyribose sugar, no alcohol in the 2' carbon. Ribose, missing an E. Ribose sugar, the 2' carbon has an alcohol group for RNA. DNA is made of A, G, C, and T nucleotides that I showed you at the beginning of the video. RNA is also made of the same A, G, C, but instead of T, uses a different nucleotide, slightly different one called uracil for U. Let's take a look. If you take a look at them carefully, there's the purines. There are 2 purines. Remember, the purines are the larger ones. I always remember it's backwards. Larger ones are smaller named, smaller ones are larger named. Purines, and there's adenine and guanine. Pyrimidines, cytosine, uracil, and thymine. Cytosine, again, both RNA and DNA use that. And uracil and thymine are, if you look at it carefully, remarkably similar. They both attach to the ribose number, deoxyribose number 1, or ribose right there, attachment point. And the only difference between them is right here. Thymine contains a CH3. And by the way, the name for a CH3 group is called methyl. So question, well, how come? Why? The difference is thymine is what we call methylated. It has an extra methyl group. I've put that as a question to you just to see if you're interested in answering that. It's on the Google Classroom. And finally on this page, this is a double helix. You imagine a ladder and you twist it up and you have the double helix structure. Very important. The connections here in the rungs are the A's and T's going together and the C's and the G's going together. They're held together by hydrogen bonds. The rails, on the other hand, the connections here, are held together by what are called phosphodiester bonds. That's a real mouthful, but think about it. You know the word ester, it's one of our functional groups, and you know phosphate, phospho. Okay, so finally, let's take a look at the DNA strand. This is the double helix unwound and made to look flat. The blue dashes, sorry. The blue dashes represent, of course, the hydrogen bonds. If you look at the beginning of the video again, I showed you how those are connected. It turns out that A and T make 3 hydrogen bonds between each other, and C and G, A and T make 2, and C and G make 3. Right? So 3 connections, 2 connections. They're about equally as strong. Whenever there's an A, there's always a T on the other side. Whenever there's a G, there's always a C on the other side. Each rung is about the same width because it's made of one larger and one smaller nucleotide. So one purine and one pyrimidine. And this is called base pairing. So always C connects with G, T connects with A. The other important structure here is not along the middle, but along the rails. So the rails are made of what are called alternating phosphate sugars. Phosphate, that's a sugar, ribose. Phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on. This is often called the phosphate sugar backbone of the molecule, and it happens on both sides. Wait a minute, this is upside down. There's a good reason for that. The two strands of double helix are anti-parallel. If you notice this one, if you look at it carefully, here's a sugar from a nucleotide. This is the phosphate that belonged to that nucleotide. I'll color that in blue. This is all one nucleotide. The nucleotide that's next to it came in like this. And so on. The connection here between the 3' carbon on this nucleotide and the phosphate attached to the 5' carbon, that right there, that's your phosphodiester bond. I'm going to be giving you an assignment where you have to research and tell me about how DNA makes copies of itself. And that'll be displayed in your Google Classroom today. But one thing that's very important to know what the chain strand orientation is. So the idea is no matter how long the side is, the last one at the top will always be a 5' carbon. And the first one at the bottom will always be a 3' carbon. So in this direction, 3' to 5'. The other chain will be opposite, 3' at the top, 5' at the bottom, and so it runs in the opposite direction. And that's why one is upside down. It's very important. It's how DNA works. And let's get into genetics because there's a lot of stuff to learn and it's all really exciting.